welcome to Cygnus and I. Next week's talk, Mary Harrington from Psychology, will ask whether any of us really knows what time it is. And today, Stan Scardellis is going to introduce our speaker. Thank you, Bruce.
It's a great pleasure whenever you can have a student return who's done some remarkable work and is continuing to do remarkable work. This is the last event of the 60th anniversary celebration of the biochemistry major at SMIT, and we're very fortunate to have Lorna Metzges, who did biochemistry but through a rather circuitous route, starting off as a music major, but it happened, okay? And so she graduated as an undergraduate in 2008, and then did a master's in my lab in 2010, went on to Yale in the prestigious molecular biophysics and biochemistry program there. Did fabulously well, I hear, from some of the faculty who are in the program, and then decided to switch fields, still staying in doing some aspects of biochemistry, but now getting a structural aspect. And so she decided to enter the rather remarkable world these days of cryo-electron microscopy, electron microscopy of liquid nitrogen temperatures, for which this year the Nobel Prize was awarded in chemistry for the development of that technique. And so she went off to learn it in John Briggs's lab, did a little bit of moving from Germany then to Cambridge, into the Medical Research Council Laboratory of Molecular Biology now in Cambridge, and soon will return to the U.S. to go to Caltech to do even more cryo-electron microscopy. But at the same time, is working on a technique called cryofluorescence, and evidently the physics of fluorescence isn't well understood at cryo temperatures, and so there are all sorts of differences that happen there I've come to learn in the last couple of days. And so it's with great pleasure that I welcome Lorna Metzges.
All right, I've accomplished the first goal of turning down the lights. So I'm really happy to be here and to tell you about my work on correlated cryofluorescence and cryo-electron microscopy, which I use to study influenza virus fusion. So I'm going to start out by just introducing some of the biology of why I'm doing this, but then I'm going to switch tasks and talk about cryoclem. So this is a really new technique. Only about 12 labs in the world currently use this, but it's growing as cryo-EM is growing. And so hopefully this will be interesting to all of you. And then I'm going to tell you a little bit about the methods development that I'm doing with the cryofluorescence. And finally, we'll put it all together, and I'll show you how you can light up influenza fusion and take some really pretty pictures. So this is influenza virus. This particular strain is Hong Kong 68, so named because it was pathogenic in Hong Kong in 1968. We're not very creative with the naming. It's what's called a polymorphic virus. So it can be long filaments, like I'm showing here, that can be several microns long, or it can be anything down to tiny spheres, about 70 nanometers in diameter. So there are all sorts of shapes that this can adopt. The one consistent thing is the proteins that it makes, that make it up. So if you look at this bottom portion, there's one protein that just sticks out like a brush all over the virus filament, and it's the hemagglutinin one, this green here. And this is the fusion machinery. It grabs on to a target membrane, and it pulls the membrane together so that the virus can enter the cells. And you can see influenza takes no chances. It's covered in this protein. So if we look at the life cycle of influenza virus, because influenza has a membrane, it has to fuse that membrane in order to enter a cell. So how this works is it comes in and it enters the cell through endocytosis. Now, this is a digestive process. So as an endosome travels into the cell, it becomes more and more acidic. And the idea is that this breaks up whatever's inside into building blocks that can then be recycled. Now, viruses like to hijack cellular processes. And so the virus is actually triggered by the acid to undergo a conformational change. And this stimulates fusion. And so if you look at, here we go. Yeah. And so right here, this fusion event, the membranes fuse together, and whatever was on the inside of the virus ends up on the outside of the endosome. And so this is how the genome goes from inside the virus into the cell. It can then go to the nucleus, you get replication, and you get more viruses coming out. So if you want to stop this process, you can target any of these steps, and those are the rectangular boxes on the slide. But the most efficient step to block would be the fusion event, because the cell would destroy the virus. All it has to do is have the time to get more and more acidic so that then the virus is chewed up. And so this is the step that we really want to learn more about, because it's the best way to block viruses. Now, why do we want to do this? I'm sure we've all had the flu at least once in our lives. You know, it's inconvenient, but it's not too terrible. But still, some people can get really sick from it, so it can cause morbidity and mortality, both in humans and in livestock. So this is a big agricultural problem as well. And influenza is so contagious that even though the probability of really severe events is small, it still actually kills a large number of people every year just because of how many people get infected. So anything we can learn to combat influenza is great. But also, a lot of viruses use acid-activated membrane fusion. And so the idea is that if we study influenza, which is relatively safe to work with, then we learn about other things like Zika and dengue that are more dangerous. And so the hope is that the lessons we learn can decrease the amount of really dangerous research that we have to perform. So influenza does membrane fusion. This has been studied for over 70 years. You'd think we would know more about it. The reason that we don't is that it's really hard to study. So fusion is a process that exists very strongly both in time and in space. So if we want to study fusion, we have to consider both temporal and spatial scales of our measurements. So in this slide, up above, I've put a time scale from nanoseconds, 10 to the minus 9, all the way up to 1 minute. Below, I have a distance scale from angstroms, which is 10 to the minus 10, all the way up to 1 meter. And in yellow, you can see that I've put in some techniques that are more common. This is not an exhaustive list, but these are some common ones. And the yellow bars correspond to sort of the general range that they cover. And again, there are exceptions, but this is sort of what's typical. So if we look at membrane fusion, the important time scale is actually a really easy one to study for us. It's in the millisecond to 1 second to 1 minute time scale that we have to worry about. And this can be studied with light microscopy, fluorimetry, a lot of techniques that we've used extensively over the years. The problem is that the distance scale is quite small. If we want to study how these proteins perform membrane fusion, we have to be able to watch the alpha helices moving. And that's a distance resolution of about 8 angstroms. So that's hard. And you can see that the two techniques that cover that distance scale are electron microscopy and crystallography. And now if you look at the temporal scale up above, neither one of those is anywhere on there. Turns out they both require completely stationary things and a lot of averaging. So you lose the time scale completely. There's no way to recover it. So if we want to study membrane fusion, we have to have a way of accessing both the time and the distance. And we need to use two techniques and correlate them. And this is what I'm going to tell you about. So I use cryo-EM, which is correlated light and electron microscopy. And I'm going to take you through the whole process from vitrifying a sample to the cryofluorescence, then to the cryo-EM, and tying it all together. So first off, why do we have to do cryo-EM? Why can't light microscopy do this for us? It has to do with something called Rayleigh's criterion. So basically, a microscope has a certain resolution limit, and this is defined here as D. And so this is defined by two parameters. Numerical aperture, which is a parameter of the microscope. It has to do with the optics that are involved. And wavelength, so the lambda here. And the wavelength is what really limits light microscopes. So the wavelength of visible light is in the hundreds of nanometers. And this is just simply what the physics is, that this is the wavelength. And the numerical aperture, we can get it to a certain level, but we can't really improve it indefinitely. So it turns out that the light requires a resolution limit of around 200 nanometers. And this is really the limit of light microscopy. It's not going to get better. You can trick your microscopes by doing various super resolution techniques, but you can't get away from this fundamental limitation of light microscopy. Now, the wavelength of electrons is picometers. And so the microscopes can get down to 1 angstrom, or even better nowadays. This is old. And so this is why we use electrons as our illumination source rather than light. Now, there's a drawback. To use electrons as an illumination source, you have to shoot them from the top of your microscope down. Electrons like to bounce off things. And so what you have to do is have a really strong vacuum. Now, if you have a really strong vacuum and you have a biological sample, all the water would instantly vaporize. You would destroy your cells, your proteins would be floating on the sides of your microscope. This doesn't work. So it used to be that people would embed their samples in plastic, but this destroys all of the high-resolution information, the details of the proteins. And so this was the Nobel Prize this year in chemistry, a guy named Jacques Dubochet, and he developed a method for cryo-microscopy, so freezing your samples so quickly that the ice doesn't have time to crystallize. And this is how you can have ice without having the electrons bouncing off of it, and they can just pass right through, and you can get good samples that preserve all those fine features, and your sample hasn't had time to react to being frozen, so it's really instantaneous. So this is why we put up with the cryo aspect. This is why we're willing to work with liquid nitrogen all the time, is because it preserves our samples. Finally, the last thing to consider with electron microscopy is that the physics of how the electrons interact with your sample is a little different than with light. And so what's happening is they're coming from the top and they're passing through, and you're looking at the angular deflections of the electrons. And what this means is that it turns out that this gives you 2D projections. It's sort of like a medical X-ray where you see everything stacked on top of each other. And so this means that what you look at can be a little deceptive. And so this is a nice example. This is filaments of influenza. And you can see this X shape right here. This is not an X-shaped virus. What's happened is that 2 filaments are like this in my field of view. They're not even touching, but they're just stacked this way, and I'm looking downward, so it looks like an X. And so this comes up again and again, where what you see isn't always completely trustworthy, because you have to consider what's happening below, on the side, above. Now, the final thing that I want you to think about is that if you have an incredible resolution, if I can blow up my sample and look at it 40,000 times magnification, my field of view is actually really tiny. So this is an electron microscopy grid. And my grids have 300 of these squares. And each one square looks like this. And what this square is it's a layer of carbon. And the carbon has holes punched in it. And what we want to do is we want to look in the middle of a hole, because remember I told you that this is a 2D projection, so I don't want to see the carbon. I've got enough trouble just seeing my sample. So I want to avoid the carbon when I look at things. Now, one square has more than 100 holes, and if I look at a resolution where I can see influenza, I'm looking at only about 1/4 of a hole. So if I wanted to look at everything on my grid, that's 300 times 100 times 4. That would take like a month. It's not going to happen. So there are 2 things that you can do to avoid this. The first is that you can have the perfect sample, completely homogeneous, spread out so that anywhere you look on your grid, it'll look exactly the same. And this is what structural people do. When they get those beautiful protein structures, they spend months just getting perfect samples that are completely homogeneous. I want to study a process that isn't going to do that. The virus looks different. Every single virus looks different. I want to study a process that isn't consistent So I can't count on just being able to pick a random spot and see something. So what I need to do is map so that I can look at this grid, this whole 3 millimeter field of view, and know where to go. And so this is the purpose of cryo-CLEM. So we use fluorescence as a way of mapping. So we can label something and have it be specific to what we're looking for, and we can find it at a much more zoomed out magnification where we can actually look at the whole grid. So how does this work? This is cryo-CLEM. We start out with our grid, and we look at each square, which is this A here. Now, if we zoom in to our fluorescence picture, this is B, we have our sample, and we also have fluorescent beads that we've put in there. And so these beads are what's called fiducial markers. We can see them in both the fluorescence and in the electron microscopy. And so they're going to help us line up what we're looking at. And in the magenta box, you can see that this is a different color than the beads. This is our sample. So we've picked a certain fluorescent signal that will look different than our fiducial markers. So once we have this picture, we grab our grid, we run across, and we take an electron microscopy image that's zoomed out. So we know in advance which squares correspond to which ones because we look at the whole grid. And then in this zoomed out picture, we can look for our beads. And so they look like this. They're little dark circles, and we can pick them out. And so again, this is the yellow circle. So we now have where the beads are in both the cryofluorescence and in the electron microscope. And then we just correlate them. So we overlay those two position maps. We rotate them, we translate them, we even stretch and compress them a little bit depending on the optics. And it ends up that we can go to something in the fluorescence and know where it should be in the electron. And so what we can finally do is go to our signal spot in the fluorescence map and move the electron microscopy stage there and take a picture. And so this is what's called a bacteriophage. It's a different kind of virus that infects bacteria. So this is cryo-CLEM. It's used as a localization tool, and it's very powerful. It's really a wonderful technique. So I want to use this now to study the influenza. Now, with influenza, I'm not just looking for the virus. I'm looking for a specific function that it's performing. And so this up here is the virus fusion events. So we start out with a virus in a pre-fusion state. It's bound to a receptor that's not being shown here. But this is the hemagglutinin. That's the protein that sticks out like a brush all over the virus. And it's sitting there. This is pH neutral. Nothing's really happening. Now, when the pH drops, this green fusion peptide right here flips up, and it embeds in the target membrane. And so we have this hemagglutinin protein that's bridging the host membrane and the virus membrane. Now, the folding energy of this protein actually wants to double back down on itself. It wants to collapse. But there's a resistance because there's an energy barrier to bending a membrane. And so these hemagglutinin proteins diffuse all along the length of the virus. And when enough of them get close together, their combined folding energies are enough to overcome that energy barrier. And so it draws the membranes close together. And this is the collapse of the intermediate. Now, fusion is not always successful. But if it is, the next step is hemifusion. And so what happens here is that you can see these proteins now are collapsed inwards, and they're pulling the two membranes together. And in this very tiny area that I'm showing with my laser pointer, we have lipid mixing of two leaflets. So the outermost leaflet of each membrane is merging. And so in that very tiny area, we have 3 membrane leaflets instead of 4. And we have lipids that are freely interchanging between the virus and the host. And this is called hemifusion. Now, again, if fusion progresses, about 45 seconds later, you get the final merging of the membranes. And so you get one continuous bilayer that encompasses both the endosome and the virus. And at that point then, as this widens, what was on the inside of the virus ends up on the outside of the endosome. So this is what the virus has to do to deliver its genome. Now, this has been well studied, and the best example is the Harrison and Van Ooyen collaboration. This was done at Harvard in 2008. And they took an influenza virus and they labeled it with 2 different fluorophores, one in the lipid membrane in green and one in the interior in red. And they looked at the fusion traces in time. And so they would watch as one membrane came down, and they had a lipid bilayer right here, and they were using turf to only look at the area near the bilayer. So as a virus would come down, they would start to see signal, they would drop the pH, and then they would see a burst of fluorescence as the dye that was in the lipid began to merge out. And then a certain time period later, there would be a pore formed right here, and the dye that was on the interior would come out and it would diffuse away. And so you would see a decrease in fluorescence. And so they were able to assign a time course based on all these changes in fluorescence. And this was very powerful because kinetics really can tell you a tremendous amount about processes. They can predict intermediates, and they could even begin to predict how many hemagglutinin molecules it required just based on the kinetics. So this was wonderful, but it's only telling us about the membrane. All the fluorescence is based on what the membrane is doing. We have no information on what the proteins are doing. And if we want to stop this process, the proteins are the machinery, so we have to stop the proteins. So we need to know what the proteins are doing. We need to know what all of the influenza proteins are doing, if they're involved in this process at all, and how we can stop it. So if we want to look at the proteins, we need cryo-EM. And this is a slice of what's called a tomogram. It's a 3-dimensional picture. It's from the Lee Lab at the University of Washington last year. And you can see it's absolutely beautiful. They have all these viruses. These are the ones that look like brushes with all the proteins sticking out. And they mix them with lipid vesicles. So these are the round, bare things. And these are the target membrane. And they're looking at the interaction between the two. And so they have this type of adhesion, or what they call HA bridging. We have this type of picture where the 2 membranes are held close together, but they still look like they're 4 separate leaflets. And that's what they call an extended interface. And they have this type of interaction where the membranes look kind of pinched together, and they call that pinching. And they also have a post-fusion, which isn't pictured here. And of course, some things they just couldn't tell what they are. Because remember, this is a 2-dimensional projection. It's hard to be completely sure what you're looking at. The issue with this is that if you try to line up what's happening with the proteins with what's happening with the fluorescence and the kinetics, it's really hard to do because we just don't have enough information. So the HA bridging could be anything prior to the collapse of the intermediate, because also something could be happening under your field of view. The pinching could be either one protein bridging the 2 bilayers, or it could be the hemifusion. And the post-fusion that they're seeing probably is actually really long after fusion occurred, because it had to get wide enough for them to be able to say, oh, this is fusion, just by their eyes. And so we can't look at the proteins and line them up with the kinetics. And this is really where the field has been stuck. So my project is to try to bridge the 2. I want to take the technique of cryo-chem, where I'm looking at fluorescence and electron microscopy, and I want to try to do a bridging so I can take the kinetic information and send it to the electron microscope. So this is how that works, just on a schematic. I have some sort of an assay that is fluorescence-based. So this is my version of that Van Ooyen with the 2 colors that I showed you. And I want to send that into the cryo-chem. So I want to take my assay and freeze it onto an electron grid. And then I want to take an EM picture of that. And hopefully the idea is that I can send that functional information into the EM so that I can look at a virus and know what stage it was in the kinetics. All right, so first we have to develop a fusion assay that's going to be compatible with electron microscopy. So the grids are a little funny. It's not quite the same as doing this in a fluorimeter. So I decided to use what's called auto-quenching fluorophores. And all this is that when 2 fluorophores or fluorescent molecules are really close together, they're dark, they're dim, they don't really give off a lot of light. When they're far apart, they're bright. Turns out a ton of fluorophores do this. I would say the majority, but it's usually not talked about because the companies that make the fluorophores don't want to admit that they're actually not that good. But we want to hijack this and actually use this to our benefit. So it's a bad product of fluorophores that we want to turn into a good thing. And so this distance dependence turns into a concentration dependence. And so if you look at a graph of intensity versus concentration, if I take something in one vesicle where the absolute number of fluorophores does not change, but I can change the concentration by adding more lipids, basically. If I have a high concentration of dye, it's dim, and then as I dilute the dye, it gets brighter and brighter. So in a fusion assay, how this works is I load a vesicle with really concentrated dye that's over here. It's quenched, it's dark. And I mix it with Influenza. Then I drop the pH, and this is what stimulates the fusion. And I get a whole mess of stuff. So there are some vesicles and some viruses that will never have fused. There are some fusion byproducts, so off-pathway intermediates that just get stuck. And then there are the fusion events. And the idea is that these fusion events, and only these fusion events, get bright because the dye that was inside of the lipid vesicle gets diluted into the virus. And so both the virus and the vesicle light up. And so if we look at this at room temperature, at pH neutral, before time 0, if I can get the laser pointer to work, there we go, it's going to be dark. And then when I drop the pH, I get this burst of fluorescence as I get things lighting up. And this is pH dependent, so I know that, yes, this is in fact due to the virus. OK, so there's a catch. This process of auto-quenching is based on something called resonant energy transfer. And it's a photophysical process. And it turns out, of course, it's been characterized at room temperature because this is 99.9% of the work that we do is at temperatures above 0 degrees when things can move. Makes sense. So we have to think a little bit about how this works to think of whether it'll even work at minus 180 degrees Celsius, which is where I work. So this is what a fluorophore looks like. It's called Alexa 488. And you can see that it's a system of conjugated aromatic rings. And all this means is that I'm going to have delocalized electrons. And the only reason that this matters and why I'm telling you is because it turns out that the energy you need to excite one of these electrons to a higher molecular orbital is the energy of visible light. And so if we shine a blue light on this molecule, we can force an electron from ground state up to a higher energy level. And it'll stay there for about 4 nanoseconds or so, and it'll lose a little bit of energy as it spins around and loses some to heat, etc. And then it wants to fall back down. Now, when it falls down, it has to get rid of the extra energy. And so this can either be through non-radiative processes, which is the black arrow, or it can give off a particle of light called a photon, so one quantum particle of light. And this is going to be at a lower energy because it lost a little bit of energy throughout this whole process. And this is what gives us excitation and emission spectra of a fluorophore. So if I look at the wavelength of light, you can see that I can excite the molecule with wavelengths in this range, and then the photon that it gives off is going to be a little bit less energy, higher wavelength, and it'll be in this range. And so you always get this little bit of a shift, and you get these tails, which come from the fact that there are substates that you can access through various motions and things like that. Now, there's another thing that a fluorophore can do to get rid of all that energy, and that is resonant energy transfer, which means it gives it to another fluorophore. And then that fluorophore has one of its electrons go up and down. And so if we think about this application, this is where the auto-quenching comes from. So if 2 fluorophores are really close together, then they can transfer their energy between them, and they stay dark because they're just passing it back and forth. And if you get just one fluorophore alone, it's going to be bright because it's just staying on this left-hand side of this Jablonski diagram. You can also do this with 2 fluorophores. This is actually more common. It's called FRET. For those of you who've heard of it, it's called a molecular ruler because it basically changes color according to the distance between the fluorophores. And the idea is the same. You excite one, and either it passes energy to the other, or it doesn't. Okay, so does this work at cryo temperatures? Do we think it will work at cryo temperatures? Well, the answer is maybe. So it turns out cryo temperatures do really weird things to fluorophores, and we completely don't understand this at all. All we know is what we see. We don't understand the physics. So the wavelength that they can excite and emit can change. It can change a lot. These excitation and emission spectra can look completely different. You can also lose the tails because you might be losing access to some of these substates. Fluorophores can go dark. Sometimes they don't work at all. And they can also lose some of their properties. So for example, some fluorophores can be switched on and off with other wavelengths of light, and that doesn't always work at cryo temperatures. So brave new world, and we know nothing about it. Okay, so do I think that this process can work at cryo temperature? Well, I don't think FRET can work, or at least if it can, it's going to be hard because we have 2 fluorophores that might respond completely differently to the cryo temperature, and the idea of resonant energy transfer is really based on both of them. So there needs to be an overlap in their spectra, the dipoles need to line up in a certain way, and so if we have 2 fluorophores behaving completely differently than expected in different ways, that's just going to be a mess. But it doesn't mean that the energy transfer won't work. It just means that the fluorophores are being a little uncooperative. So if we're just using 1 fluorophore, then maybe this will still work because even if it changes a lot, its integral properties may remain constant. Okay, so I developed a test, and I'm not going to go through all the fluorophores I've tried. I'm not going to put you through that. But this is a proof of concept test. And so what I'm doing is I'm taking 2 different sizes of lipid vesicles, so sizes that I can see in the electron microscope, and I'm putting different concentrations of dye in them. So one of them should be quenched and dark, and one of them should be bright. Now, I've chosen these sizes and concentrations carefully because these 2 vesicle sizes and concentrations should have the same actual number of fluorophores. So the concentrations are different because one is bigger than the other, but the actual number of fluorophores is the same. And so if I look at a histogram, where I look at size versus brightness, I should see if quenching is not successful, and if I failed and my postdoc is going nowhere, then I would see this kind of a plot, where everything would have different sizes because that is something I designed, but they would all have the same brightness because they all have the same number of fluorophores. Now, if I've been smart about choosing my fluorophore, then I should see successful quenching. And this should give me 2 populations. So I should have something that's big and bright and something that's small and dark. And I should also have a lot of noise, which I've drawn in here because these things are never perfect. So if I look at my results, unfortunately you can't see all of the spots here, but this is a cryofluorescence image, and this is the histogram. So I'm looking at the size, so spot radius, versus the brightness, the raw intensity. And each one of these events corresponds to one of these dots. And you can see I have a population of large bright things and a population of small dark things. So I was very happy. This is, as far as I know, the first time anyone has proven that RET works at this temperature. And it also means that we can begin to use the fluorescence at cryo temperatures to tell us more than just localization. We're not just labeling something to find it. We're going to label a function. So I wanted to detect hemifusion. This is when the lipids just start to mix. And this is the fluorescence signal that I'm telling you about. So if I look at my fusion graph here, I have my time course. So this, again, was pH neutral, where everything is dark. And then I acidify it. So I drop the pH, and I see this burst of fluorescence as I get fusion. And I freeze it right here. And so I expect, again, to have a mixture of things on my grid. I should have dark vesicles that have a lot of dye but just haven't fused. I'll have viruses. And somewhere, I should have bright things that should be the fusion events. And so this is what my grid looks like in the cryofluorescence. This is one grid square. And remember, this has 100 holes in that, and there are 300 of these squares. And if I zoom in on just one of these tiny spots, and I do the correlation with the cryo-clem, and I take a picture right here, you can see I'm finding fusion events. And so this is interaction between the lipid vesicle and the viruses. And some of these I wouldn't have necessarily known were fusion events. It's just that they're bright. And so I've been taken to them, and I can take a picture. So this is very well and good. I can find what I'm looking for, and I can search for a function rather than just a molecule. But it can also do something even more powerful, and this is what's really excited me anyway. So this is what's called a tomogram. So I've basically done a three-dimensional reconstruction of my field of view, and I'm moving in Z. And so you're going to see it slice in and out. And this long thing is one filament of influenza, and these are the vesicles. And so I want you to look at this movie and just try to see if you can decide what you're looking at. What are all these different interactions? And so you can see where vesicles get pulled, and so they're not perfect spheres anymore. And somewhere up here is going to be some pinching events. And right here, there's going to be a vesicle that actually wraps all around like half of the virus. It just is hugging it. So we have all these different images. So can we use fluorescence to tell us what's going on? So this is the cryofluorescence image of this. And again, these are squares of the grid. And so the actual field of view is very tiny spots in here. The green are fiducial markers. These are the beads that are not my sample. And the blue that you see on these grid bars here, that would be the color that we would expect from a fusion event. You can see there's no blue. This is an adhesion-arrested population. So all of those interactions we were looking at, none of them are lipid mixing, even though we would have said by eye that they were. They're actually not. And so with this, we can start to look at the different conformations of the virus, different proteins, and start to assign them based on the kinetics rather than just based on what we see, which can be really deceptive. So this is the hope anyway. Eventually, the idea is that we'll be able to make movies with cryo-EM based on the kinetics, because we can timestamp with fluorescence every event. So the future, first thing we have to do is improve our microscopes. Turns out cryofluorescence microscopes are really awful. The optics are just horrendous. And so there's a lot that you can't change. It's just the fact that microscopes were not designed for this temperature. But there's a lot we can do. And so I've built an addition to our microscope with the help of some people at Heidelberg. And basically what they had to do was raise this entire thing off of the table because we were getting condensation from the temperatures. And so it was an amazing feat of engineering. It's like 50 pounds worth of optics on that pedestal. Hope it never comes down. And so what we're starting to do now is beginning to look at the protein conformations. And so what I'm really interested in is the matrix protein of influenza. And this is the one that gives influenza its shape. And so it wraps around in sort of like strands all along the underside of the membrane. And this is what gives the influenza mechanical resistance. Because otherwise, I mean, you're coughing constantly when you have the flu, and it's coming from deep in your lungs. And so the virus would be destroyed if it didn't have some way of resisting all of those forces. The issue is that if the virus resists forces, it'll also resist membrane bending, and so you won't get fusion. So the virus has to get rid of the matrix protein layer at some point in order to allow fusion to occur. And so we want to figure out when this happens. And so if I look at influenza at pH neutral, this is another slice of a tomogram. This is the matrix layer right here. You can see these stripes. So this has a very specific spacing, and this is here on this histogram. The distance between these stripes is the distance between M1 strands. And at pH neutral, it has a very defined distance. Now, if I move to an acidic pH where I would get fusion, this is actually the acidic pH, the filaments begin to look funny. They can twist, they can double back on themselves, they can even fuse with each other, which is really a problem. And so you can get these weird things that look like this, or you can get straighter filaments. But that movement means that they have to be able to bend their membranes. And so if we look at the strand spacing, probably the most impressive thing is that it doesn't change that much, actually. But what is changing is the standard deviation of the spacing. And so this means that basically when you can move the strands apart, it gives you just enough room to bend that membrane. And so we think that this is actually the first event in allowing a membrane to fuse, is just having that little bit of flexibility in the strand spacing. All right, so there's a lot more that we're trying to do with this, but this is very much a work in progress. So I'm going to wrap up and just thank my lab. So I'm a postdoc, which means that I have a great boss, and I have a lot of lab mates, and we all train each other and work together. And I also had wonderful training from facility staff. So electron microscopes take a lot of maintenance. They're really hard to learn to use, takes about a year. And so facility staff play a big role in this, and so these guys were great. And then my collaborators at EMBL Heidelberg, who helped me figure out how to build optics on a pedestal, and my funding sources. And thank you for your attention. And if anyone has any questions, please. Liquid helium.
Liquid nitrogen or liquid helium? Helium is colder.
Yes. So there do exist a few liquid helium electron microscopes. The hope was that this would actually, I think, decrease damage from the electrons. In reality, it didn't seem to benefit. And so, I don't think there's too much understanding of why that is. But I think that the benefits from liquid helium probably are there, but I don't think our microscopes actually are good enough to take advantage of them, because we're still limited very much by our lenses. So electron microscopy lenses are actually coils, and so it's not easy to bend electrons. And so, yeah, I think our microscopes just aren't good enough. Yeah. Yeah.
Great talk. Thanks so much. I was wondering if you could tell me a little bit about the sort of scalability of this. So in terms of the correlation piece, like let's say you're labeling whatever protein you're interested in, and then you correlate it to that given image, producing that location. Can you move that to like quantification? So like how many things are, how many viruses have this protein at this stage of kinetics?
Yes.
And is that even like possible, or is that like?
So depending on the size scale you're looking at, it's possible. So my proof of concept, I'm actually trying to quantify right now. And I've scaled this up to handle one whole grid square at a time. And so I can get, about 100, 200 events that I can say are one thing or another. With the viruses, the issue there is that you really need to be at a very high magnification, and your field of view is just very small. So the issue is really taking enough EM images, and the holdup is microscope time. So I think that the hope is automation. So we can currently automate to collect images all night long without us being there. And I use the fluorescence to guide the automation. And so I can set up all my positions and have it go to those locations during the night. The issue is just having enough microscope time to actually get to all the positions. Yeah. I'm sorry, I can't hear. Yes. Yeah. Not a special way. The smaller ones are more homogeneous than the larger ones. And so you can measure the vesicle size by something called dynamic light scattering. And I did this. And so my smaller vesicles have a standard deviation of a little less than 50 nanometers. But the larger ones, you're right, they're a lot less homogeneous. And this is actually where the cryo-EM comes in, because I used the EM to tell me how big they were. Yeah. Then.
I have a question about why you zeroed in. Maybe you explained this at the very beginning, but why did you choose to work on influenza virus?
So I wanted to work with viruses, and it turns out that there aren't a lot of strong human pathogens that you can work with under biosafety level 2 conditions. So this was sort of the natural choice for that. HIV, dengue, Zika, all of the really interesting ones are much more dangerous. And I can't put them on the microscope without people yelling at me.
I have a question about the proteins involved in the coating and the ones that when you said that you observed that they may trigger actually the opening before fusion.
Yes.
Are those solved with are the structures known?
Yes.
They are in the PDB?
The hemagglutinin, the pre-fusion and post-fusion structures are in the PDB. However, they are just the ectodomain, the part that is soluble and you can express independently. And so what we don't have is the transmembrane domain and the cytoplasmic tail. So there's a cytoplasmic tail that probably interacts with the matrix protein, and we have no structural information about that. Thank you.